Hi guys, welcome back to my channel. This is Dr. Rajeshwar from YR PharmaTube. In the previous video, we have learnt the phase 1 hydrolysis reactions of drug metabolism. If you did not watch the video, hit the I button on top right of this video to watch it. To watch the other topics of drug metabolism and other subjects of pharmaceutical sciences, click on the links given in the description box below this video. In this lesson, we shall try to understand the hydration reaction, miscellaneous phase 1 reactions, and a summary of phase 1 drug metabolism. At the end of this video, you will be able to understand the hydration type of phase 1 reactions, miscellaneous phase 1 reactions, and summary of phase 1 drug metabolism. Hydration reaction. In chemistry, a hydration reaction is a chemical addition reaction in which a substance combines with water. In other words, hydration is a chemical reaction where a proton and a hydroxyl group are added to a compound. In most of the cases, in organic chemistry, water is added to an unsaturated substrate, which is usually an alkene or an alkyne. This type of reaction is employed industrially to produce ethanol, isopropanol, and butane 2-ol. The general scheme for hydration reaction is illustrated in the figure. For example, industrial ethanol is produced by using hydration reaction of ethene, which is an alkene. Hydration can be regarded as a specialized form of hydrolysis reaction where water is added to the compound without causing the compound to dissociate. In drug metabolism, hydrolase enzymes catalyze the addition of water molecule to a variety of functional groups. Thus, epoxide hydrolase enzyme hydrates epoxides to yield trans dihydrodiols. This reaction is documented for many aromatic compounds and epoxides of olefins. Here, a molecule of water has been added to the substrate without loss of a molecular fragment, hence the use of the term hydration. The pre-carcinogenic polycyclic hydrocarbon epoxides, in particular, undergo this reaction, for example, benzopyrene 4,5 epoxide. The reaction forms a transdiol and depending on the substrate may be stereospecific. Miscellaneous phase 1 reactions. Many other reactions which cannot be classified into the groups discussed in the previous videos such as the oxidation, reduction and hydrolysis. Some other reactions have been proposed as possible routes of the metabolism for specific drugs. A list of some of these reactions is given in the table here. Summary of phase 1 drug metabolism. As discussed in the previous videos of drug metabolism, virtually every possible chemical reaction that a compound can undergo can be catalyzed by the drug metabolizing enzyme systems. In most cases, the final product contains a chemically reactive functional group such as hydroxy, amino, thiol, carboxylic acid, etc. And thus, as we shall see in the future videos, it is in the correct chemical state to be acted upon by the phase 2 or conjugative enzymes. Indeed, it is recognized that the main function of phase 1 metabolism is to prepare the compound for phase 2 metabolism and not to prepare the drug for excretion. Phase 2 is usually the true detoxification of drugs and gives products that are generally water soluble and easily excreted. It is also appreciated that many drugs can undergo a number of the reactions listed previously. Indeed, some drugs can pass along many of the routes of metabolism described above. The importance of a particular pathway varies with many factors, most of which are described in the previous videos, and it is obviously very difficult to predict the metabolism of a drug from the data given above. Computer-based expert systems for the prediction of routes of drug metabolism are also available and are becoming more accurate as data is added into them, but it is still not possible to fully predict the metabolism of a particular compound from its structure. However, molecular graphical techniques are giving a greater insight into the structure of the enzyme active sites and how substrates fit into the site. Such work should yield a model of substrate enzyme interactions and thus a better understanding of the relationship between substrate structure and its metabolism. Phase 1 metabolic reactions introduce new and polar functional groups into the molecule which can produce certain changes in the drug molecule, such as decreased pharmacological activity, increased pharmacological activity, increased toxicity, for example, carcinogenesis, mutagenesis, and cytotoxicity, and altered pharmacological activity. Drugs exhibiting increased activity or activity different from the parent drug usually undergo further metabolism and conjugation, residing on deactivation and excretion of the inactive conjugates. In conclusion, the phase 1 biotransformation reaction introduces a new functional group into the drug molecule, modifies an existing functional group, or exposes a functional group on the drug molecule. Certain phase 1 metabolites hereafter are excreted and or undergo phase 2 or conjugation reactions. This is the list of references followed for the lecture. That's all in this video. The phase 1 hydration reactions, miscellaneous phase 1 reactions, and the summary of phase 1 reactions of drug metabolism. In the next lesson, we will discuss about the phase 2, that is the conjugation reactions of drug metabolism. Till then, never stop learning and never stop watching my videos. Thank you for watching this video.